Okay, so thank you for inviting me. And before I begin, I would like to acknowledge the contributions of my students and postdocs who have been integral to this work, especially Jesse Gomez, who has recently finished his PhD in my lab, but was formerly an undergraduate here with Brad at Dartmouth, and Vaidee Natu, who is a postdoc working with me, and formerly she was a graduate student with Alice O'Toole. And the work I'll describe today, thank you, Marge, for a very inspiring beginning to this workshop. What I'll tell you today is about development of the face recognition system in humans and during childhood and not during infancy. So faces are really important for everyday social interactions, as there is a key source of information we use to recognize other faces. So while we can recognize faces in a fraction of a second, face recognition is an ability that develops across development. And as shown in these three independent studies, adults are better at face recognition compared to children. Thus, face recognition is an ability that develops from childhood to adulthood. So in order to study face recognition mechanisms in the brain, the first thing that we need to do is to localize face-selective regions in the human brain. And we typically do that with a localizer experiment in which we show subjects a variety of stimuli, including faces. And we've developed this localizer that has 5 domains of stimuli. There are 2 categories per domain. And we spend time calibrating stimuli across domains for low-level properties such as contrast, luminance, and similarity across images. So to localize face-selective regions in the brain, we'll typically find regions that or voxels that respond more strongly to faces compared to other stimuli. I'm going to show you today processes in the ventral aspect of the temporal lobe or ventral temporal cortex in humans. And typically we see a set of 3 face-selective clusters on the ventral surface, 1 patch in the inferior occipital gyrus that I'm going to refer to it as IOG faces, 1 patch in the posterior fusiform that we are going to refer to as P-fuse faces, and 1 patch about 1 and 1.5 centimeters more anterior that I'm going to refer to as M-fuse faces. And a characteristic of all of these regions is that they show higher responses to faces compared to other stimuli. And importantly, the selectivity to faces is maintained across stimulus formats and when controlling for low-level low-level features across stimuli. Critically, these regions are causally involved in face perception, as electrical brain stimulation of P-fuse and M-fuse patches results in the specific deficits in the perception of faces. Now, we can localize these face-selective regions within individual participants, and we can find these face-selective patches even in our youngest 5- and 6-year-old subjects. And the spatial relations of these face-selective patches in children is similar as to adults in respect to other category selective regions, for example, place-selective regions shown here in yellow. So I will argue that the face processing system in the brain is an excellent model system to investigate the development of specialization in the brain and its relationship to behavior. And in my talk today, I'm going to address 2 big picture questions. First, what brain mechanisms develop from childhood to adulthood? And second, how do these brain mechanisms or these brain developments lead to better face recognition behavior? So in considering these big picture questions, we address in our research 2 competing developmental theories of the brain called pruning and growth. So pruning postulates that brain development is associated with pruning of unnecessary synapses and connections. And it's based on neuroanatomical data. For example, this is data from a classic study by Pascal Rakik and colleagues that shows that after some initial increase in synapses from 4 months old to adulthood, the number of synapses and connection decreases in a macaque V1. And the researchers have also postulated that the functional consequence of pruning is reduced responses to irrelevant stimuli. So if you're in a face-selective patch, it predicts reduced responses to things that are non-faces. A competing hypothesis of brain development is that of growth. And this idea is that development is associated actually with increases in the sizes of dendritic arbors, synapses, and connections. And this is also based on neuroanatomical data, but from a different part of cortex, an IT cortex that Marge described previously. So this is data from a more recent study by Elston and Fujita, and they recorded the sizes of dendritic arbor in area TE, which is in the anterior portion of IT, and both the dendritic arbors and the number of synapses increased significantly with development. And the idea is that this increase in arborization increases pooling, and the functional consequence of that might be increased responses to the relevant stimuli, for example, increased responses to faces in a face-selective patch. So I'm going to describe a set of experiments that we have examined both the structural and functional predictions of both the pruning and growth hypothesis. And as I'm going to describe these brain findings, I'm also going to evaluate if these developments have behavioral consequences. So the studies that I'm going to describe to you involve school-aged children between the ages of 5 to 12 and young adults between the ages of 22 and 28. In order to obtain good quality data in our children, we train them in a mock scanner to stay still and fixate on our stimuli. We acclimate them into the scanner environment by first collecting anatomical MRIs on them, and then they participate in a series of experimental sessions in which we collect fMRI data. And in addition, we also collect quantitative MRI or qMRI, which I'll expand on later on. Then we do behavioral measurements of face recognition, primarily outside the scanner. So the data I'm going to show you is a lot of data. We have multimodal data of more than 250 hours of collections on these participants. Critically, we analyze each data in the participant's native brain space. We do not spatially smooth our data. We use independent data to localize brain areas and to evaluate function and instruction. And importantly, we test and verify that there are no general between group differences in the quality of the measurements, in the subject's motion, or in the signal-to-noise ratio. So let's get back to our developmental hypothesis. So considering the structural hypothesis, pruning is associated with decreases in cortical tissue, and growth predicts increases in cortical tissue. Now, with in vivo measurements that we have available to us today, we cannot do cellular level measurements like has been done in the monkey brain. However, what we can do is leverage the development of new quantitative MRI sequences to examine structural changes in vivo in our human participants. So what is quantitative MRI? So the qMRI sequence that I'm going to describe was developed at Stanford by Aviv Metzer, Brian Wendell, and colleagues. And basically, the sequence is designed to remove RF bias and give you a measurement that has real numbers and units that describes cortical tissue. So the measurement I'm going to describe today is T1 relaxation time. It's in units of seconds. And the idea is it's analogous to something like taking somebody's temperature. It has a real value, and you can compare across individuals. So to give, why are we particularly interested in T1 is that T1 is inversely proportional to macromolecular tissue volume, or MTV, as well as the physiochemical environment of the tissue. So the more tissue you have, and especially if it has lipids and cholesterol, such as in myelin, the shorter your T1 measurement will be. So this is an example coronal slice showing a T1 map in a participant. And if you look at different tissue structure, for example, if you consider a ventricle, the ventricle has mostly water in it. There's not a lot of tissue for the protons to exchange energy with. It has a very long relaxation time, around 4 seconds. In contrast, if you look at the white matter is heavily myelinated. And as a consequence, the protons relax rapidly, and we have relaxation times of less than 1 second. So how can we use this T1 measurement to look at our structural predictions? So remember, pruning predicts that from childhood to adulthood, there's going to be less tissue because of less connections, less synapses. So basically, compared to a child tissue, the adult tissue has less tissue. There's less stuff for the protons to exchange energy with. And what we predict is an increase in T1 relaxation time from childhood to adulthood. In contrast, growth predicts the opposite, is that from childhood to adulthood, there's going to be more tissue. As there's going to be more tissue in the adults in the child brain, the T1 relaxation time should decrease from childhood to adulthood. So basically what we're going to do is we're going to measure T1 relaxation time and see if it increases or decreases across development. So we scan our subjects. We first localize our category selective regions, for example, face and place selective regions in each individual, and then extract from these regions the T1 relaxation time and compare across age groups. So this graph shows average T1 relaxation time in children and adults from p-fused faces, and we have a significant reduction in the T1 relaxation time from childhood to adulthood. And this is true not only when you look at the mean response, but if you look at the distribution of T1 relaxation times in adults compared to children's are shifted to the left, meaning overall lower relaxation time. It is interesting that this development seems to be specific to the face-selective regions, because if we look in the same participant in the place-selective region, that's 2 centimeters more medially, we don't see any development either in the mean T1 relaxation time or the distribution of T1 values. So these measurements are consistent with the predicting of the pruning hypothesis because T1 has decreased from childhood to adulthood. So does structural development impact brain function? So one metric of brain function is looking at how selective a brain region is to its preferred stimulus. And you can think of functional selectivity as a metric of saying how good this region is at discriminating the stimulus from another stimulus. So in each participant, we measure with independent data as a selectivity or as a preference for faces, and we see if there's a relationship between preference to faces and T1 relaxation time in our p-fused faces. So each dot in this graph is a participant. The childs are in circles, the adults are in diamonds, and we see a significant negative correlation between face selectivity in p-fused faces and T1 relaxation time. That means that participants with shorter relaxation times have higher face selectivity. And this relationship remains significant when we factor out age from this correlation. It's also interesting that in the place-selective cortex, where we don't find a developmental effect of T1, we also do not find a correlation between place selectivity and T1 relaxation time. So these data indicate that the structural and functional developments are linked, and then we ask if these developments affect face recognition abilities. So to assess face recognition abilities, we use a test that has been developed by Brad Duchesne and colleagues called the Cambridge Face Recognition Test. And this basically is a test that evaluates how good people are at discriminating and remembering faces. We use a variant of the task that has child faces because we don't want our children to be at floor behavior. And so for each subject, we have a measure of how good they are at recognizing faces, and we test if there's a correlation between recognition memory for faces as assessed by, say, CFMT compared to the T1 relaxation time. And again, we find that there is a significant correlation between, or significant negative correlation between mean T1 relaxation time in p-fused faces and recognition memory. So the shorter relaxation time, the higher the face recognition memory is. And again, this effect is significant when you partial out age. And importantly, this effect is specific to the face-selective regions, because if you correlate face recognition memory with T1 in the place-selective cortex, it's not correlated. And if you correlate place recognition memory with T1 relaxation in the place-selective cortex, it's also not correlated. So we were very excited about these results because they suggest that there is a tripartite relationship between structural, functional, and behavioral development. But this also raises a host of new questions. And specifically, the kind of questions that it raises is what compartments in the tissue might be developing from childhood to adulthood. Is it more neurons that are in the adult brain as compared to the child brain? Maybe the adult brain has more myelin in it. Maybe there are increases in the dendritic arbors and the number of synapses. The problem is that addressing any of these questions really requires you to do histology. So in order to test these hypotheses related to specific compartments, we need to do histological measurements that are done at the microarchitecture level. And this raises two complications when you want to do experiments with people, is that in order to do these microarchitectural measurements, you need access to postmortem tissue. postmortem tissue is really not accessible in children. So this is one issue. And the second issue is we have to have a method that we can register functional regions in an in vivo preparation to anatomy or anatomical landmarks in a postmortem tissue. So luckily, we've found some roundabouts across this, and I wanted to show you some data that would give us some insight about what might be the underlying compartments of tissues that might be development. So one of the things that comes to our aid is that in work that I've done with Kevin Weiner, we found that there is actually very reproducible structural functional coupling in the ventral temporal cortex in adults. And what I mean here is that macroanatomical landmarks are very good at predicting where functional regions are in the human brain. So this is an inflated brain of the human being. This is the ventral stream, ventral temporal cortex, and this is the fusiform gyrus. And within the fusiform gyrus, there's a sulcus called the mid-fusiform sulcus, or MFS, and it bisects the fusiform longitudinally into medial and lateral sides. And what we find is that this sulcus, even though it's small, it's a good predictor of where face-selective patches are, and specifically they tend to align to anterior and posterior end and always lie lateral to the sulcus. So the idea is that if you can identify the sulcus either in an anatomical preparation, or you can do cortex-based alignment from a functional region in one brain to another brain, you might be able to predict from anatomical landmarks alone where this face-selective region might be, even in a postmortem brain. A second thing that we found is that this is not only for face-selective regions, but anatomy or macroanatomical landmarks can predict other regions. For example, if you're interested in place-selective cortex, all you have to do is find the intersection between a sulcus within the lingual gyrus. It's called the ALS, or anterior lingual sulcus, and the collateral sulcus, and if you find the intersection between these sulci, there's a high probability that you're going to land on place-selective voxels. So again, these macroanatomical landmarks can be identified in a postmortem preparation, and we can use them to make prediction about where face and place-selective cortex might be. Another thing that we can do is use an adult postmortem tissue to test some of our hypotheses. Another observation from our data is the following. If you look at children, the T1 properties of face and place-selective cortex are not differentiated. So they're on top of each other. However, if you look at adults, you can see that the T1 properties of face-selective and place-selective cortex, shown in yellow, are differentiated in that the face-selective cortex has lower T1 values than the place-selective cortex. So it's basically the result of development generates a structural differentiation in the temporal lobe. So what we can do is we can take an adult brain and ask questions. For example, since we found lower T1 in face and place-selective cortex, can this be explained by some macroanatomical features of place-selective cortex? For example, is this because there are more neurons in face-selective cortex compared to place-selective cortex? So one of the reasons that we're interested in these questions is that other work that we've done in collaboration with Katrine Amutz and Carl Zillis found a nice coupling between cytoarchitectonic partitions of the brain and face-selective regions. So this is the group map of the face and place-selective regions, and we found that there's good co-registration between functional regions and cytoarchitectonic regions. So P-fused faces overlaps with cytoarchitectonic regions called FG2, and place-selective cortex overlaps with the different cytoarchitectonic regions, FG3. And what defines the cytoarchitectonic regions is exactly the differences in the cell density across layers. So what we can do is measure what Carl and Katrine call the gray level index, or the distribution of the cell density across layers, and test if there are differences between face and place-selective cortex. So for example, if we think that lower T1 is because of more cells in face-selective cortex, you would expect that the GLI in face-selective cortex would be higher than the GLI in place-selective cortex. So this is data from 10 postmortem brains, but contrary to our hypothesis, we found the opposite. The cell density in the place-selective cortex overlapping FG3 is higher than the face-selective cortex in FG2. So while we have cytoarchitectonic differences between these areas, this cannot really explain the lower T1 values in place selective cortex. So this suggests that what is developing is probably something in the neuropil rather than neurogenesis or cells being born in this piece of tissue. So it's not the number of cells that's developing. What about another tissue compartment, for example, myelin? So you might ask yourself, like, is there myelin in human ventral temporal cortex? What do we know about it? And can myelin really explain these T1 developments that we saw? So we forged another collaboration with researchers in the Max Planck Institute in Leipzig with Evgenia Kirillina and Nicholas Weisskopf. And basically in these preparations, what we've taken is postmortem tissue. We have 5 brains, and we stained them instead for a myelin. So this shows an example histological section from a postmortem brain. This is a fusiform gyrus. This is an MFS. This is a collateral sulcus. And this brain is stained with silver staining that tracks myelin. So the darker it is the more myelin there is in the tissue. And you can see that white matter is very heavily myelinated. It's black. But if you look at the cortical sheet, and I saw this image, I say, wow, look, the cortex has an extensive amount of myelin, especially in the deep layers that are close to the white matter. And what we can do is that we can quantify this. So for example, we can measure the optical density, which is a marker of myelin, from the PL surface to the white matter. And you can see that as you go deeper into cortex, you have a higher level of myelin. So we can also do this in place selective cortex. And again, we measure the optical density from the PL surface into the white matter. And consistent with our hypothesis, face-selective regions have higher myelin concentrations than place-selective regions, which might explain this reduced T1 in the face-selective cortex than place-selective cortex. But can this really explain our data? So what we can do is actually measure myelin, but we can do computer simulation to see how much myelin would need to grow in tissue from childhood to adulthood to account these differences that we measure in T1. So what we did is another thing that we can measure from our quantitative MRI is macromolecular tissue volume, or basically the amount of non-water in the voxel. And we wanted to look at this metric because it's directly correlated with the amount of lipids and myelin in the tissue. And our developmental measurements suggest that there's about a 13% increase in macromolecular tissue volume in the child face-selective cortex compared to the adults. So what we did in our simulation, we simulated how much would need the volume of the myelin sheaths to grow to account for this 13% increase in macromolecular tissue volume. And so Jesse did a range of simulations. We varied the initial axon diameter as well as the density of the axons within a voxel. And what we found out from these simulations is that in order to have a 13% increase in macromolecular tissue volume, the axons would have to grow to some enormous sizes such that the axon's diameter, the myelin sheath's diameter is much bigger than the axon diameter. And this doesn't even look like any axons in the brain. So these are axons that are heavily myelinated in the corpus callosum. And you can see that the axon diameter is much bigger than the myelin sheath. So what we think that is happening across development in terms, it's more about the method of elimination than actually having a direct measurement because it's so difficult. But what we think is probably myelin is increasing in this tissue from childhood to adulthood, but that is not enough to explain the T1 differences. And it's probably the case that additional things that are growing in the tissue, and in particular, if we leverage the data from Elston and Fujita, it's probably increases in the sizes of the dendrites and the number of synapses that are contributing to this reduction in T1. And we're really interested in this idea of increase of arborization because we think that it would affect the neurons that actually wait or pull information into a given neuron. And that might explain why we have changes in selectivity from childhood to adulthood. Okay, so far our structural measurements support this hypothesis that development is involved with increases in cortical tissue. Let's go back and think about what are the functional consequences of development. So if we look at the predictions of the pruning and growth hypothesis, to remind you, pruning predicts decreases in the responses to preferred stimuli. So if we're looking at responses in face-selective patch, we expect them to be higher for faces and other stimuli. But what pruning predicts is that what happens with development is a decrease in responses to non-faces without changes in responses to the preferred stimulus. In contrast, the growth hypothesis predicts the opposite outcome, in which we should see a significant increase in the responses to faces with no big changes in the responses to non-faces. This is a very easy hypothesis to test, because all what we need to do is identify face-selective voxels in children and adults, use independent data to extract signals, and just see what comes out. And this is precisely what we did, and this is an example from M-fused faces. The blue shows a response in children to these stimuli. The orange shows a response in adults to these stimuli. And you can see that there's a significant increase in the responsiveness to faces, but we don't see significant differences in the responsiveness to non-faces. And this is consistent, again, with the prediction of the growth hypothesis. So far we have functional and structural data that are consistent with the growth hypothesis. However, you might ask, why would higher cortical responses in face-selective regions make you better at recognizing faces? So we hypothesized that another facet of functional development might be increased neural sensitivity to face identity. And we predict that increased neural sensitivity might lead to increased perceptual discriminability and consequently lead to better face recognition ability. So how are we going to measure neural sensitivity to face identity? So what we're going to do is leverage this idea of fMRI adaptation and use that to measure face sensitivity and neural sensitivity to faces. So a large body of research has shown that when you show the same face, you get reduced responses in face-selective regions as compared to showing different faces. And this is because of neural adaptation to repetition of the same face. A lot of research in adults has shown that this adaptation is dependent on the similarity among faces. So the more similar faces are, the bigger adaptation you will have. So what we can do is we can parametrically vary the similarity among faces and measure the response level with fMRI. And then we can measure this relationship and quantify the slope of this line. So I'm going to define the slope of this line relating response level to face dissimilarity as neural sensitivity to faces. And what our developmental prediction is that if neural sensitivity increases across development, the slope of this line should be higher in adults as compared to children. So Vaide designed a really nice fMRI, a parametric fMRI adaptation experiment, and she morphed a source face to 6 different target faces, and then she varied parametrically how much she will weigh the source and target face. And in different blocks, we have different source and target faces as well as different weighting of the stimuli. So just to give you a gist of the stimuli, this is an example of 3 faces that are identical. This is from 40% morph level, and this is 100%. These are really different faces in the real environment. So we're going to localize face selective regions in each participant and then measure responses in this region using this fMRI adaptation experiment. So first I'm going to show you the mean responses in M-fused faces. So again, this x-axis shows you the morphing level, the y-axis shows you the response. Blue are children and orange are adults. And we can see that in both children and adults, we see this adaptation, that responses are lower for identical compared to different faces. And on average, you can see that the slope seems to be steeper in adults than in children. So what we do is we fit this line in each individual and compare the slopes of these lines across age groups. So this is data from both P-fused faces and M-fused faces. Again, the orange are adults, the blue are children. And this is when they look at adult faces. And consistent with the developmental hypothesis, we see that there's higher slopes in adults than in children. This is consistent with this increases in neural sensitivity. The same phenomena happens when both age groups look at pictures of child faces, but we see some evidence for what's called an own age effect. So you can see that in adults in M-fused faces, the neural sensitivity is higher when they look at adult faces, compared to child faces. And in P-fuse faces, when children look at child faces, they have higher neural sensitivity compared to adult faces. And this suggests that maybe both overall experience but also frequent experience with stimuli of a particular type might affect this neural sensitivity. So we next tested if this neural sensitivity has some perceptual consequences. So outside the scanner, we measure how well people perceptually discriminate between these faces. So we have now a metric of perceptual discriminability and a metric of neural discriminability, and we want to test if they're linked. So again, in these plots, each participant is a dot. They're color-coded by their ages. And what we see is that in both face-selective regions, the higher the neural sensitivity, the better people are at discriminating among faces perceptually. And this relationship is specific to face-selective regions in two ways. First of all, if you look at this correlation compared to the overall amplitude of response to faces, this is not correlated within face-selective regions. You can measure adaptation in other parts of the brain, for example, in LO, and adaptation in LO does not predict perceptual discriminability of faces. So this suggests this development of neural sensitivity in face-selective regions predicts perceptual discriminability. Another interesting finding from our data is that these functional changes also impact the informativeness of distributed responses in ventral temporal cortex. So here we did a decoding experiment in which we took the entire ventral temporal cortex and saw how well we can decode from this distributed activity if people are looking at a category of faces, either child faces or adult faces. And you can see that overall adults are better, the decoding is better from adult data than children data, but interestingly, this improvement seems to be particularly for adult faces. And this suggests again that not just overall experience with faces, but perhaps adults have more experience with adult faces that leads to this better discriminability of the classifier. So, so far we have a lot of evidence in favor of the growth hypothesis because we also see increases not only in responses to the preferred stimuli, but also increases in the neural sensitivity to face identity, as well as increases in the informativeness of distributed responses to faces. Which leads us to the last set of questions that we wanted to examine, and this is really how is all this shaped by experience? So as visual scientists, we want to start at the most basic experience that people will have, and this is very much in line with Marge's work that she showed earlier, and that is like looking behavior. And the reason that you want to look at looking behavior because this is how you get input about your stimulus. And also in the domain of face recognition, there's a lot of evidence that people have very stereotypic way of looking at faces. So in particular, when people are asked to recognize faces, people tend to look at the center of the face. So this is an example from our data during a recognition memory test outside the scanner. And this shows you the fixation density across 11 adults. And you can see that people look around the center of the face when they're asked to recognize faces. And we think this is interesting because it's tied to receptive fields in face-selective regions. So in other experiments, we have measured population receptive field, or the receptive field of a voxel within face-selective regions. These receptive fields, their centers are shown here in the red. They show this contralateral bias. But you can see that the receptive fields in the face-selective region are big, and they almost always cover the fovea. So what we think is this is the reason that people look at the center of faces is that you're putting your computational resources, your receptive field, and the informative regions of the face is that have the features that are informative for face recognition. So what happens in kids? If you look at the looking behavior in children in the same kind of task in a recognition memory test, they don't look randomly in the face. They kind of look at the center of the face also like adult. But you can see in this example that they seem to have a much more, they have a somewhat shifted behavior, is that they're shifting their fixation upwards and rightwards compared to adults. So the question is this developmental difference in looking behavior a result of differences in the population receptive field and the visual field coverage in the face-selective regions from childhood to adulthood. So what we wanted to do is measure PRFs in children and see how they differ, or if they differ between children and adults. So what we do is we ran this retinotopic mapping experiment that's been adapted to be kid-friendly. So instead of a boring fixation, we have a spaceship. The spaceship has an alien called Dot that has to go to its planet. And when the spaceship runs out of fuel, it turns red, and you have to press a button. So children like this task. They like to have perfect scores. They're really good at it. Also, during the scanning, we have eye tracking, so we are sure that our children can fixate on the stimulus. And as they're doing this dot task, we have a checkerboard bar that's sweeping across the visual field, and we're going to be mapping receptive fields across the entire visual cortex. So first of all, what I wanted to tell you is that the retinotopic maps, at least by the age of 5-year-olds, are very similar across children and adults, both qualitatively and quantitatively. So this is an example of an eccentricity map. So basically red is fovea, and then blue and green are more periphery. And this is an example medial view of a child brain. This is an example medial view of an adult brain. And you can see this in the child brain, the two eccentricity representation. One in the occipital lobe, going from posterior to anterior, from fovea to periphery. This is in a child, this is in an adult. And then you can see the second eccentricity representation in the ventral cortex, ventral temporal cortex, going from the inferior to superior, from fovea to periphery. So basically, the children have intact retinotopic maps, and they look very similar to adults. If you look at PRF properties, they also don't differ between children and adults. And if you put all of these together, you can look at the visual field coverage of all receptive fields in a given area. This shows the visual field coverage in the left V1 of children. This is in the left V1 of adults. And you get this typical hemifield representation in both children and adults. And this retinotopic organization of the ventral retinotopic regions, both early and intermediate, seems to be already developed by the age of 5 years. So basically, in any parameters, either qualitative or quantitative, we couldn't find differences in PRFs or visual field coverage across children and adults. So what happens in face-selective regions? This is now the visual coverage map in the PFU's faces, average across 14 children. Consistent with what I've showed you before, we have this contralateral bias. Right hemisphere covers the left visual field. We also have evidence for this foveal bias, is that there is a higher density of coverage of the visual field by PRFs around the center. However, interestingly and different from these lower level retinotopic regions, we see a development in the visual field coverage in face-selective regions. Specifically, what we see is that we have an increase in the coverage of the visual field, so it extends more into the ipsilateral visual field and to the periphery in adults. And additionally, the foveal bias seemed to increase. As you can see, that there's just more red here in the adults compared to children. And we can quantify this by measuring the center of mass of the visual field in each participant and comparing across age groups. So basically, lower values on this graph would indicate that the foveal bias increases because the center of mass is closer to the fovea. And it's interesting that we see this development happening in the right hemisphere, but we don't see this development happening in the left hemisphere. So the foveal bias is specifically increased in the right hemisphere. I'm not going to show you here, but in the same paper, we have data from the word selective cortex. And in the word selective cortex, we see the opposite effect, that we have increases in the foveal bias in the left hemisphere, but not in the right hemisphere. So what does this mean, this change in the visual field coverage? So suppose you're a child and you're presented with the following face, and now you're just going to fixate on the center. If you're going to fixate on the center, what you're going to do is you're going to put the resources of your face-selective regions, the visual field coverage, in a place that doesn't really have a lot of facial features that would be informative for your face recognition. So what you should do instead is shift your fixation rightwards and upwards, because if you do that, now you're putting on your computational resources on the area in the image that actually has information for face recognition. So to test this hypothesis, we actually measured outside the scanner people looking behavior at faces or free viewing faces while they're asked to recognize them. And then the images are jittered, so we looked at each face if there are differences in the way that children and adults look at faces. So this vector shows the differences in the vectors of fixations of children versus adults. And it's interesting that we find a significant and consistent bias that children tend to look upward and rightwards on faces compared to adults. And this is consistent with them putting their visual field coverage on the face in the place that there is informative information for recognition. So to conclude, what brain mechanisms develop from childhood to adulthood? I've shown you data that shows that development of face perception involves both structural and functional growth rather than pruning of cortical tissue. Now, I'm not saying that pruning doesn't happen, but what I'm trying to say is that pruning is not a ubiquitous mechanism. So while pruning might happen early in infancy in V1, it doesn't mean that happens all over the brain and across different times during developments. So basically, our research suggests that there are additional developmental mechanisms that happen in later periods during development and also might differ across different parts of the brain. How do these developments lead to better face recognition behavior? We have shown that cortical tissue growth in face-selective regions is coupled with both increased functional responses to faces and increased neural sensitivity to faces. Consequently, this leads to better perceptual discrimination and recognition of faces. So why might this growth happen? So in terms of a visual stimulus, faces are a really particular difficult stimulus. After all, you need to distinguish between faces that have the same features and they're arranged in the same spatial configuration. So when you're an infant, your social circle is small, and maybe the faces of your environments are very different because your grandmother's face is very different from your brother's face. But as you grow, both your social circle really expands significantly, and this requires you to distinguish and recognize hundreds and maybe thousands of faces. I hope you're going to tell us tomorrow. So we speculate that these behavioral necessities require a refinement of the underlying brain mechanism, especially those that are associated with foveal vision, and that is what leads to this prolonged development of the face recognition system. And as in a final note, I would also like to propose that our results underscore the importance of combining anatomical, functional, and behavioral measurements in the same participants to understand brain mechanisms of development and their impact on behavior. And while I'm really interested in the visual system, not only on faces, but I think these tools are in generally going to be important in the field to looking at the development in any cortical system across the lifespan. So thank you very much for your attention. Yes?
So when we looked at responses to faces and objects in what would become face domains, we saw a decrease in non-preferred, so essentially what I called pruning. But I'm skeptical of our results because the signal change, you know, with the going from the baby to the adult, the signal actually gets smaller in the adult. And we had issues with babies looking at the screen and so on. But I kind of remember Jessica Cantlin and Stanislas Dehaney had a paper where they came to the conclusion there was pruning in the formation of maybe text domains. I forget what it was. So why should it, what do you think is going on?
Okay, so I've looked at this for more than 10 years. So we've looked at it initially in 2007 with Golliger-Golari. And what we did in that study, and I showed you data from variety studies, but we've found this result now in 3 separate studies with different stimuli and different participants. The result is very consistent that what we see is increases in the response to faces and not really changes to non-faces. And in Golizhe's original experiment that we published in 2007, what she did is she took the adult face-selective region because it's bigger than in children, and she looked at the penumbra. So basically, the regions that are going to become face-selective. And it's similar to your result in that if you look at this region in kids, it's not differentiated. So we have similar responses to face as an object, but what we see is happening in the adults is actually higher responses to faces. And our result is similar to other results coming from the Keiji Tanaka lab. There's a paper from Kobataka and Tanaka from 1998. And this is just they had like adult monkeys in the lab and they kept showing them stimulus. And they reported a result that like finding a neuron that's tuned to any of the stimulus that the monkey was exposed was much higher probability after years that they've been exposed to the stimulus than before. So I really think that the selectivity is moving towards experience stimulus. The paper that you're referring to is a paper from Jessica Cantlin and Stan Dehan from 2010, and they didn't indeed claim pruning. If you look at their data, you could decide whatever you want to call it. It's not, because there are big differences in the overall signal between children and adults, and it's hard to know like what is what. So I've done this experiment 3 times. I get the same results, so.
So one thing we didn't do, and I maybe think we should have, was normalize to something like V1. But did you ever think of doing that? Like how, because we have such a big change in the overall signal in
so
the babies.
I don't have a justification to do this in my data because we get very similar signals from children and adults for the non-faces. And if you look at other, because we do look at all categories. I mean, this is a face conference. I'm showing you just data for faces. But we have looked at other category selective regions. Maybe I have the slide here. So if you look at maybe LO or place selective cortex, LO by the age of 7 is fully developed. We don't find signal differences. In the place selective cortex, results vary across groups, but I'm absolutely sure that it's fully developed by adolescents, where in face-selective regions we still see it develop during adolescence. So we don't really have this overall decrease. Again, we're looking at children, not at infants. Maybe if we looked at infants, it was a different story. But there isn't like this overall signal difference across age groups.
Yeah, I have a few comments, questions. So this seems consistent with some work Isabelle Gauthier did with. She found that cortical thinning corresponded to increased face recognition ability, if I remember right. And that's consistent with the stuff underneath the neurons, kind of pushing them up against, squeezing them. Another comment, Ben Ballas has found this cool result that people from small towns have worse face recognition behavior than performance than people from cities. And I wonder if you could look at kids that are homeschooled versus kids that go to daycare and see if there's a difference in their performance depending on exposure to the number of faces.
So, yeah, I mean, these are all good points, and it would be a really interesting study to do. The kids that we have done are all coming from the Palo Alto and neighboring school districts, so they seem like the typical, I don't know, American suburban population. We have an ethnically diverse population in California, so it's not just seeing like one race.
Even in Berkeley or Stanford, you must have some right-wing homeschooled kids.
They probably are. We haven't specifically targeted this group, so it's a good idea for an experiment. We just haven't done them. Okay. In terms of cortical thinning, the work that I'm doing with Vide is exactly looking at the cortical thinning, and we actually see that myelin really predicts well cortical thinning. So we're really sure that it's not about pruning. We're really sure that it's just about becoming more myelinated.
I'm agreeing with you.
Okay, good.
Yeah. Third question. So I was wondering, in your face-looking results, what was the methodology? Because when Janet Chow and I did this work, we started off the face and then let them saccade to the face, and in that case, you get a leftward bias. They saccade to the left side of the nose, right about here, and then they move rightward. I wonder if you started off the face, or you just presented the face and let them start looking at it directly.
So the way that the experiment is there's a stimulus, and we have actually in the experiment many stimuli from various categories. So they have faces, they have places, they have bodies, they have words. So it all comes interleaved, and basically there's an encoding phase that they're doing a one-back task. So they're seeing the stimuli and saying if it immediately repeats. Then we have a 3-minute gap in which they actually see a rotating checkerboard, because we use that to train them on the fixation task. And then subsequently, they get a surprise recognition memory test. So in the recognition memory test, there's a fixation in the center. So between stimuli, there's going to be a gray screen. And then randomly, a stimulus from any of these categories are going to appear. And then they review the stimulus. And it's self-paced till they decide if they recognize it.
Is the stimulus at the center of gaze already?
So the stimulus is not always center. So this is basically, there's some jitter in the sizes of the faces and in the location, but we don't really have a face that's like out there, all in the periphery. So they're somewhat around the center, but they're jittered. So you'll have to move your eyes to the facial features, because they're not going to be always exactly in the same spot.
Yeah, so when we did it, you fixated the center, and then the face would either appear above or below fixation. In that case, you get this leftward bias,
And
which is the opposite of what you seem to get.
Well, I think it's not the opposite, because again, if you look at if you really believe in the superiority of the right hemisphere for face recognition, you do get this. The right hemisphere represents a visual field in the left more than the right, and that would prompt you to saccade more leftward. That would be consistent with.
No. If you saccade left, then you're actually putting less of the face to the right hemisphere.
Not on the left eye of the stimulus, you saccade leftwards. That would be weird then.
Yeah, it is weird.
Yeah, so we don't get that, no.
That's what I've been wondering about for several years, is because you're putting actually less of the face into the right hemisphere when you do that.
Wow.
And all I can think of is there's some balance of resources.
So there's a lot of research. Bruno has done research on eye movement, and there's all this old research from Pelfrey and stuff. This is very typical of where people look. They typically look here, and if there's a bias, it's actually going to be on the left eye of the stimulus, which would be consistent with our results. So maybe something about putting them in the upper or lower visual field makes a difference in your experiment.
That makes sense. So if I can jump on this, the right hemisphere needs less.
Yeah.
It needs less resources. So if anything, you could explain. I mean, I know you can explain it both ways, right?
I don't agree with that.
I don't see a contradiction with a right hemisphere superiority.
Yeah. Okay, let's let somebody else, and then I'll get back to you.
Please, the next one. Yes.
Hi. So I just wanted to follow up on the exposure ideas that you started talking about at the end. Do you have any sense of what's driving individual differences in ability? You could imagine it's just the number of faces that you're exposed to, or the diversity of those faces, the amount of time you spend looking at faces, propensity to attend to faces, that kind of thing. Is any of these in the lead, or are they all on the table at the moment?
I think these are all really, really good questions, and I don't have any answer. And I'm going to take Marge's move, and I'm going to go back to the comfort of vision science and talk about receptive fields. So we have another study. It's in bioRxiv. It's a study that I did several years ago with postdoc Nathan Woodhoff. And in this case, we took people who are developmental postprognostics. So these are adults, and they're really bad at face recognition. They don't have any brain lesions or anything like that. And we measured PRF in the ventral stream and visual field coverage, kind of like this. We have 7 developmental postprognostics and 15 typical adults. So in adults, the coverage looks the same. In the postprognostics, the early visual field coverage looks the same. But what happens in the face-selective region is that they have really, really small coverage in face-selective cortex. So one possible explanation of an individual difference is that they don't integrate information across a big span of the face. So basically, an idea is that the receptive field might be the basis for spatial integrations that might be necessary for face perception. So this is one alternative. The other kind of evidence that would support the experience hypothesis is things like this kind of result, this kind of result, which shows an own age bias. So basically here it shows that you have higher neural sensitivity in adults when they're looking at adult faces and child faces. And in children, you see the opposites, that they have higher sensitivity. It's overall lower than adults, but it's still a little better for child faces and adult faces. So this suggests that it might be, one is your environment, because children, like adults, like young adults, especially the ones that are in college, don't see a lot of kids on a daily basis. And second of all, it also might suggest that the social saliency might also affect the need to individuate. So if you're a kid, you really care if you're playing with Johnny or Mary, but like there are these, like that person's mom, it's like a category. It's not like a person, it doesn't even have a name, right? So that also suggests that your social saliency, and if you really need to individuate that person, would affect these kind of developments. But right now, this is speculation. I don't have direct evidence to show that.
Thanks.
Let's.
So I like these T1 relaxation results. And you must have a map. I mean, you show a couple of areas, but you must have a cortical map, because what about the other face areas, like the STS areas, the anterior temporal areas, inferior frontal areas?
Oh, we haven't gone that far. So I can tell you what happens. I don't have a map I can show you. It's in the supplementary data of our paper. If you look at the ventral stream, you see that the development of T1 happens not only in the face-selective region, it extends to the occipital temporal sulcus. You'll see these things also happening in the word-selective cortex, and it's happening more in the lateral VTC than the medial VTC.
Did you go anterior to that?
What?
Did you go anterior and lateral? So STS and anterior temporal.
So I have the data.
Yeah, I know you do.
And it takes, and I haven't looked at it. That's the short answer. So we have new postdocs, new graduate students. We have tons of data. We're actually collected longitudinal data on all of this. So we're in the, we've collected all of year 2. We're starting to collect year 3. So I'm very happy you asked this question. And maybe in a couple of years, I could actually answer you what happens in the STS. I don't know. Maybe Brad, Galit, and then Marge, yes.
So you found that relationship between T1 relaxation time and face recognition performance, but you didn't find anything for places when you looked in the place area. Do you have any thoughts on why there would be a relationship in the face area, but not in other areas?
So one thing it might be that the dynamic range is smaller. So because in the face area we have this development, we have both dynamic range in terms of the T1 values and in the recognition values. So this is why we can get the correlation. We do have a dynamic range in terms of the T1 values in the place-selective cortex. It's not as clamped, but it doesn't seem to be correlated. And maybe this is because the recognition performance has a lower dynamic range. We did look at other cortices, but so again, the T1 in place-selective cortex doesn't predict face recognition ability. You look at a nearby word-selective region, it also does not predict the face recognition ability. So it does suggest this idea of this activity-dependent plasticity, that if this cortex is specifically trained for a task, you get a specific change that is correlated with that task at hand. Again, it's not direct evidence, but at least it's consistent with that idea.
So about the eye movement data, we actually see that there are large individual differences in eye movement. So there are like mouth fixators and eye fixators and nose. I mean, everybody's looking somewhere, and it's very consistent. We found that it was consistent like 18 months later. So do you look at individual differences? I mean, within the kids or the adults, and whether that correlates with your measures of ERF?
So the answer is we haven't looked. So this is the average across 11 adults and
right.
12
So when
children.
you do the average, it's there. But if you look at each one of them, everybody's looking somewhere else.
So I just feel that we would need to collect more than 11 participants to start looking at individual differences.
Mm-hmm.
So we can look at it, and we haven't looked at this in this particular data set. The place that we've looked at it was a prosprognostic, but on them we don't have eye movements data. So
yeah, because it's very reliable and it's
So that would be really interesting to look at.
Okay.
Yeah,
All right.
I agree with you.
And in the beginning, you showed the correlation with the T1, I think, with face selectivity.
Yeah.
Do you do that also across voxels? Because again, you did it across individual subjects, but it should correlate also across voxels if you look within subjects.
We did it at some point, and I don't.
I mean, would you think?
I think I just don't remember the results. This is
one. I was wondering, not the recognition, the selectivity.
This is this one.
The beginning.
Yeah.
Right.
So this is across participants.
Right, across voxels within participant. I mean,
I don't know. We've looked at it a long time ago, and I just can't remember what was the result.
But it should, right? I mean, you would expect that. it should work across voxels. If that's
think
a
it's a good-
property of the
Yeah, so the question is what you really think about what predicts the selectivity and what predicts the recognition. So if you think it's happening at a voxel, like I wouldn't commit to the thought that a voxel would predict your face recognition ability,
Sure.
but the selectivity might be very local. But I just, we've looked at a lot of things that like in individual subjects, like for example, curvature, because you might be worried that the curvature would be correlated with a T1, and then we looked at that definitely is not correlated. So things like about the physical anatomy in the brain with an individual subject are not good predictor. They don't show this correlation.
Okay.
So, but I just don't, we did it so long ago, I just
Okay.
don't remember what was the outcome. You had a last question, Bruno?
Myelin?
Yes.
The Myelin maps. So, like decades ago, there was some idea that the periphery, well, it was shown that you process things faster in your periphery than in your central visual field. And I thought there was some early studies on myelination in V1 that it was more myelinated in the periphery than in the center. And now you've got the opposite, which is the face down IT is more myelinated in the center than the periphery, right?
Yeah, so we're actually
Think
looking.
about that. And if
I'm-
you looked at V1,
So we're looking at that. So I have a new graduate student. Her name is Dawn Fincy. She also was here with Brad at Dartmouth, and this is exactly what she looks at right now. We're actually looking at the projections. We can track the fibers from V1 to the face areas. Jim, we're also looking at fibers to the STS. So I have answers to both of you. And we're really interested in this idea because we have other results from our lab that suggest that the temporal processing is different between the ventral regions and the lateral regions, and the temporal processing is much faster. The temporal capacity is much faster in the STS than the face regions. We think myelin might be a way to speed up processing. So we're actually really, at these moments, we're looking at it. So I think it's a really good hypothesis, and I'll tell you once I know. Bruno, you had a last question?
Very brief. I wanted to get back to the difference between the left and the right hemisphere, because you came to it at the very end of the talk with the fixation. But did you find any difference in terms of the increased sensitivity to face recognition and in terms of your T1 signal between the left and the right hemisphere? I mean, there's such an overwhelming difference between the right and the left in terms of prosopagnosia, including the transient prosopagnosia that you know very well with electrical stimulation. And here you didn't show any difference between the two, so I would be curious.
So we've done a lot of, any developmental measures that I've done, because we have the data, we're looking at both hemispheres. And consistently, this is the result we find. We find development happens in the face-selective regions, both in function and instruction in both the right and left hemisphere, but the effect size is much bigger in the right hemisphere than the left hemisphere. So all the data I showed you here was from the right hemisphere because the developmental effect is much more pronounced. In the left hemisphere, the effect is smaller, so sometimes it turns to be statistically significant and sometimes it doesn't. But depending how you think about statistics, you can make your own decision. So it shows the same trend, is just not as robust. So that is consistent with this right lateralization. The interesting factoid for you is that if you look at the size of face-selective regions in children, they're actually similar across the right and left. There's no differentiation. And so what happens across development is the size, extent of face selectivity increases, but it increases more in the right hemisphere than the left hemisphere. The data that I wanted to show you from the same paper is about words, because I think it's interesting because it shows this material by hemispheric differences, and it ties this idea that the faces and words might be fighting on foveal resources, that you mentioned briefly. So this is in the same participants. We also looked at the word selective cortex, and in the word selective cortex, it's slightly lateral to face selective cortex and the OTS, and it's supposed to show a left hemisphere domination because of the contribution of language area. So while we see that the foveal bias increases in the face areas in the right hemisphere, we see that the foveal bias actually increases in the left hemisphere, in the left visual word form area. And you can see that this pattern is also mirrored in the fixation patterns, that the children have a consistent bias, but a bias leftward. So this is a example of fixation. We use pseudo words, so if you can't understand what this means, it's okay. So this is basically adults, when they look at words, they also fixate centrally, and kids' fixations are leftwards and upwards. And you can see this consistent bias here. And if I show you the word selective cortex results, you can see that the foveal bias in the left hemisphere increases from childhood to adulthood. So this is kind of interesting because this is the clearest case that I've seen in my own research that shows this hemispheric lateralization that is material.
Did you try this with, I'm over here.
Okay, sorry. There's an echo here and I can't place your voice, sorry. Yes.
Did you try that with Hebrew? Because
I've asked my friend Michal Ben Shachar to do this because I don't have a lot of Israeli learning to read kids.
Because when I look at a word, I always tend to look at the beginning of the word because that's more informative about the word than the end of the word. And in Hebrew, the beginning of the word would be on the opposite side.
So I'm hoping that Michal will do this research, and she has a lot of kids in Israel that learn to read Hebrew, which is right to left.